This video will be analyzing the phylogeny of hemichordates by Alyssa Weisblatt, L. Eisner, and Helen Kim. First things first, let's get to know some facts about it. Hemichordate live in marine environments. There are two ways that they eat. The first is called suspension feeding, where they catch small particles and small animals.
Like this?
Not quite. They catch them as they drift by. The second way that they feed is called deposit feeding, where they consume sediment and digest organic matter while burrowing in the sand. The body of the hemichordate is split into three parts: the proboscis, collar, and trunk. Lastly, there are two classes of hemichordate: Antropnusta, which is known as the acorn worm, and Pterobrachia. Continuing on with the topic of the classes of hemichordates, here are some basic descriptors of them. The Antropnus are solitary and free-living and look like this. The Pterobrachia, on the other hand, are colonial tube-dwelling and look like this. Just kidding, it looks like this. Anyway, it was previously thought that hemichordates were paraphyletic, since Antropnus appear to be more closely related to chordates and pterobrachs were more closely related to echinoderms, given differences in larval and adult morphological characteristics. However, molecular data have been more recently shown that hemichordates are monophyletic and that pterobrachs may even be derived within Antropnus. Now, the name hemichordate reflects the idea that hemichordates share morphological characteristics with chordates, but molecular data has shown that hemichordates and echinoderms are sister groups. Their name therefore literally means half chordate. Some examples of this are that they share the echinoderm trait of larval morphology and share the pharyngeal gill pores of chordates, but then lack the dorsal postanal tail of chordates.
Three major groups are recognized within deuterozomes at the phylum level: 1 hemichordates, 2 echinoderms, and 3 chordates. Early deuterosome evolution remains somewhat murky due to poor fossil records and a large morphological disparity between the body plans of each phyla. Blair and Hedges' study proposes a phylogenetic framework of deuterosome evolution based on a molecular analysis of amino acids to estimate divergence times. Based on their study, deuterosomes originated prior to 900 million years ago, and the first divergence between chordates and the clade-containing echinoderms and hemichordates occurred around 896 million years ago. The split between echinoderms and hemichordates is estimated to have occurred at 876 million years ago. The phylogenetic relationships among deuterosome animals have been debated for many years and continues to be debated. There are three proposed theories regarding the relationship of chordates, hemichordates, and echinoderms. The traditional hypothesis places hemichordates on the chordate lineage, which is supported by shared pharyngeal slits and notochord-like structures. The second hypothesis groups hemichordates and echinoderms together as sister taxa, forming the Ambulacralia clade. This theory is based on the similar larval forms of the two phyla. The calcichordate hypothesis groups echinoderms and chordates together to the exclusion of the hemichordates. This theory is based on fossil evidence, which is interpreted to be chordates possessing echinoderm-like features. Based on recent molecular phylogenetic studies of mitochondrial and nuclear DNA sequences, there is strong evidence supporting the Hemichordata and echinoderm clade and the rejections of the traditional and calcichordate theories. The figure shown is an updated phylogenetic tree based on 18S rDNA data, which places Hemichordata as sister taxa to echinoderms. A hemichordate and echinoderm clade implies two things. First, that any homologous features shared between hemichordates and chordates would have been present in the last common ancestor of deuterosomes. And second, that the echinoderm adult body plan characters, which include radial symmetry, a water vascular system, an endoskeleton, and ring-shaped nervous system, were not present in the common ancestral deuterosomes, and that those characters are likely part of an autopromorphic transformation in the echinoderm lineage after it diverged from the hemichordate lineage. The molecular phylogeny of Hemichordata groups are continuing to be revised as further molecular and morphological data is being collected.
We constructed two phylogenetic trees based on the morphological characteristics and the 16S ribosomal DNA data of 8 taxa. On the left is the 16S tree, which shows the relationship among taxa based on nucleotide sequences from ribosomal DNA. On the right is the morphological tree, which shows the relationship among the taxa based on 16 morphological characteristics, including symmetry, digestive system, nervous system, sex determination, reproductive strategy, and lack or presence of eyes. The similarities between the two trees are not hard to see. For one, the outgroup on both trees is Porifera, and Cnidaria is also a distant relative of Hemichordata in both trees. Both trees are also similar in that echinoderms, annelids, Hemichordata, mollusca, arthropods, and chordata can be grouped into a clade. However, the differences between the two phylogenetic trees lies within this clade. One of the most significant differences between the two trees is that the 16S tree indicates that chordates and hemichordates are sister taxa and the most closely related. However, the morphological tree indicates that Annelida and Hemichordata are most closely related. With respect to morphological phylogeny, it makes sense that hemichordates are so distantly related to chordates. For example, neither hemichordates nor the annelid we dissected had appendages, whereas the perch, which was the chordate we dissected, did. Chordates and annelids had other similarities in their morphological phylogenies, such as a nerve cord, a hydrostatic skeleton, and lack of eyes.
We would like to thank Dr. Willett, Dr. Dowd, Tanya, our TA Candace, and the LMU Biology Department for allowing us to dismember bodies for research.